Thank you for being here. Today I will be presenting what is my master project. The title is Genetic Analysis of Estrus Expression Traits in Lactating Dairy Cows. So, why we care about estrus expression? So we know that high-producing cows, they have shorter and less intense estruses. We know that these shorter and less intense estruses makes the heat detection more challenging for farm personnel. And the lower estrus detection on the farm causes a lack of synchronization between the artificial insemination and the cow receptivity, which results in lower pregnancy rates. The current solution are synchronization protocols, and they work really, really great. But we are applying this protocol in a context of increasing concerns and awareness from consumers and food processors. So we, as geneticians, we believe that we can improve things at the genetic level. So what we want to do, we want to improve the estrus expression through selective breeding for two reasons. The reason number one will be because if you have a population of animals that they are able to increase the expression of the estrus, then you will increase the detection, you will increase the insemination rate, and you will increase the pregnancy rate. And second reason, we already know that those animals that they have higher expression, they also have higher fertility. So this correlation, we could use this correlation for improve the fertility of the population. Again, at a genetic level that it gives a long-standing effect. So this is the objective that we have from the genetics perspective. So the first step in this project was to characterize a estrus expression, estrus event. We do not know when the animal is physiologically ovulating, but what we do know is the activity and the rumination of the animal. So we are working with high-frequency data. Every 2 hours, we have a data point of activity that is in the plot below and of rumination that is in the plot in the top. So this is information from a single animal, and the data is already chronologically sorted, and you can see clearly how you identify, just by visually speaking, these peaks in activity. Are these peaks in activity the ones that we strongly believe, okay, they are estrus events, and are the peaks that we need to characterize in order to select for animals that they have more standing estrus expression. To do the characterization, basically we are working with a sliding window technique. We are framing, let's see if this works. Okay, we are defining two windows. One is a narrow window that we use to summarize the current activity and rumination of the animal, and the second window, which is wider, we use this window for constructing a threshold. And you can imagine this window iterating every 2 hours and scanning all the information from a single animal and searching whenever the current activity is overweighting the threshold for activity and when the current rumination is underweighting the threshold for rumination. And whenever this happens, with the computer, you should flag a 1. So for each animal, you will have all the sorted data, and you will have a vector of 0, 0, 0, 0, 0, 1, 1, 1, 0, 0, 0. And whenever you have the 1, okay, you can identify a change in behavior. Next, okay, you identify, and next what you do, you go, zoom in, and you extract characteristics from this event that you are able to identify, pinpoint with a computer. So we are talking about 3 vectors. One vector will be as a function of the duration of the behavioral change. The other will be the intensity in the change in activity, and the third one will be the intensity in the decrease in rumination. So the duration of the estrus will be a function of the number of consecutive positive flags that you are flagging in a given animal. The intensity in activity is a function of the area under the curve that is given by the threshold from the past and the current activity. And it ends up being a single vector because basically you sum up all these rectangles at the area under the curve, and then you divide by the duration, and you then end up with a single vector that the greater the vector is because it's more steep, the change. And the same logic applies for rumination. So for each event in each animal, you pinpoint this outstanding change in behavior, and you extract three vectors. So something that we need to do, otherwise people from especially physiology, will hate me if I say that I only take conclusions using a computer. We need to validate stuff using biology. And to do this is that we bring all the reproductive events and we model how is the probability of this reproductive event ending up in a pregnancy success. So we control for a bunch of fixed effects that are linked or attached to the reproductive event, and then we add the information of the estrus event. For example, this reproductive event is matching an estrus event, so we have a linear model. The response variable is 0, 1, so we are working with a logistic regression, and we evaluate how is the impact of the estrus expression over the likelihood of pregnancy success. And this is what I am going to show now. We do a logistic regression, we fit the model, and then we evaluate how is the association between the expression of a estrus, no or yes, at the insemination time, on the likelihood of pregnancy success. So keeping everything else constant, working in Holstein animals, in lactation number 1, using sexed semen, when the insemination is made by a given technician, in a given year season, like you keep everything else constant, and you see that the change in the likelihood of pregnancy success for breeding 1 when you perform the insemination without having estrus at that time versus when you do exactly the same but matching a hit event, we expect an increment of plus 10% in the probabilities of pregnancy success. And the vertical lines that you will see in all this presentation are 95% confidence interval. So whenever you see that the vertical lines are not overlapping, so we have something that is significant. This plot that you see here is exactly the same plot that is here, and the only thing that I am showing here are different factors affecting the probability of pregnancy success. This is breeding 1, breeding 2, breeding 3. This is lactation number 1, lactation number 2, sexed semen, lactation 3 or more conventional, 3 or more beef semen. So in all the scenarios, we always observe an increment in the probability of pregnancy success of plus 10% or plus 16%. It depends, and in most of the scenarios, the confidence interval, they do not overlap. So what I'm showing here is that these behavioral changes that we can flag with a computer are in fact biologically meaningful. And this is important because at the end of the day, this is the trade that we are proposing. So we are doing exactly the same for duration. The higher the duration, the higher the probability of pregnancy success, even after accounting for other predictors. And now we do want to know how is the heritability of these traits, because we are showing that these traits are closely connected with the probabilities of pregnancy success. So this that we can flag with a computer, in fact, is kind of a window to a biological process that it makes sense and is economically profitable also. So this is the same plot that before, but with more information. So let's talk about heritability. The first trait that we are analyzing is estrus expression as 0 or 1 in a window of 21 days in milk. So we segment the data. We go from 11 days in milk to 31 days in milk, and we ask to all our cows which one is showing estrus between 11 to 31 days in milk. Some of them will not show estrus, so will be 0, and others, it will show estrus, so will be 1. So the trait is a binary trait, 0, 1, and is always estrus expression within 21 days in milk. So we take the heritability of this single trait. And then what you do, you take this same window and you slide plus 1 day, and you repeat the process, plus 1 day, and you repeat the process, until reach the limit of until reach the window that go from 90 to 110 days in milk. If you do that and you systematically extract the heritability, then you get this plot. So each data point is the point estimate for the heritability. This is the days in milk. This is the value of the heritability. We see that the highest value is around 0.2, which is a repro trait. So 0.2 for a repro trait is kind of a lot. And the vertical lines is the 95% confidence interval. What is interesting is that this is most of the time, on average, the voluntary waiting period that ends up ends around 60, 50 days in milk. Here we have the TAMI, and these are repeating cows. The ones that are not getting pregnant here, they do have a second insemination around here. So something that I also find academically interesting is that see how we drop the heritability to values close to 0 when we perform an intervention in the environment that is not in the model. So we are changing, we are making a change in the variable, the response variable. My model is the same always, so the heritability gets diluted. So the implications of this is that if in the future we were to use this trait for selecting for higher expression, hence higher fertility, we should focus in the voluntary waiting period. And we should probably ignore or not pay so much attention after the voluntary waiting period because we have more noise. Finally, before we were talking about heritability within 21 days in milk. This is exactly the same, but now we are going from whatever is going on between 11 to 70 days in milk. So the first that you can do, you can count for each animal the number of events, estruses, that you can detect in this interval. And also you can extract the average duration, activity strength, and rumination strength. And you use that as a trait. So this is the 4 traits: detection count, duration, activity strength, rumination strength. This is average. And all of them, they do have the vertical lines are 95 confidence interval. And to make life simpler, we can say that any of them is different than 0.2. So 0.2 is the heritability that is equal across all the traits that I am showing here. And finally, everybody asks normally about the genetic correlation between my trait and milk production, because they say there is a negative genetic correlation. If we improve for estrus expression, then we will reduce in milk production. And in fact, yes, we have a negative genetic correlation. So these are the same traits that I showed before, and this is the genetic correlation with 3 or 5 standardized milk production. So we see this is the 0, and we see that overall the genetic correlation is almost minus 0.3 for all of them. So minus 0.3 is not different among traits. So this is my presentation. This was my master's. So the takeaway points are that we can phenotype a behavior using just a script. And this is super important because this means that this methodology we can scale up for large farms. So this is one point. Other point, the phenotype that we can extract, they are biologically meaningful. They are heritable, but they are negatively correlated with milk production. And this project, the full support for this project was provided by the Wisconsin Dairy Innovation Hub. I am open to questions.
All right, questions for Agustín?
Yeah, thank you. Very nice presentation.
Thank you.
So that particular phenotype, this estrus expression trait,
yes.
So it's basically a phenotype, right?
Yes, it is.
So this suggests that this trait might be controlled by something, like might be a gene, a set of genes, or a big group of genes. I don't know. But what is the biology behind this particular trait? So why there are some cows that are expressing or that are displaying higher rates of estrus expression compared to others? What is the speculation? What is controlling this particular
It is beyond
trait?
the hypothesis that I test. Like when I have a project, I have an hypothesis, and I test the hypothesis. So this hypothesis, whatever question I may have about that, is a new hypothesis. So it will be unprofessional to me to speculate about that because I don't know. Sorry for if the answer is frustrating, but it is what it is. I don't know why.
We have time for one more quick question. One more quick question if anybody has one. All right. If not, let's thank Agustín.